Good day, everyone. I hope you're ready because today we're unlocking one of the coolest secrets of life: how your cells read DNA instructions to build you piece by piece. Our topic is codons and amino acids. Before we start, let's look at what you should be able to do by the end of this lesson. Please listen carefully because I'm about to read the learning objectives. At the end of this lesson, the student will be able to define the genetic code, codons, and amino acids. Identify the start codon and stop codon. Describe the features of the genetic code. Demonstrate how to use a codon chart to translate an mRNA sequence. Explain how mutations can affect the genetic code. Alright, let's get started. Let's warm up with a question: who can remind me what DNA is? Just shout an answer if you have an idea. Perfect! DNA is like a massive recipe book, but here's the twist. DNA doesn't build proteins directly. Instead, it shares a code. It's like a secret language. It tells your cells how to build every protein in your body. This is called the genetic code. Without it, you wouldn't have enzymes to digest food, pigments in your eyes, or even the keratin in your hair. So, this code is a life's translator. To understand the code, we need a quick refresher. Let's see who remembers their nucleotides. So, what are the 4 bases in DNA? And what about the RNA? Which base replaces thymine? Great! So DNA bases are A, U, C, and G. DNA stays safe in the nucleus, but when it's time to build proteins, the instructions are copied into mRNA or messenger RNA. This mRNA leaves the nucleus and goes to the cytoplasm to be translated, then to the ribosomes, the cell's factory. So, how does ribosome read the instructions? The answer: codons. A codon is a sequence of 3 RNA bases. Each codon tells the ribosome which amino acid to add next. Why 3? Well, with 4 possible bases and reading them 3 at a time, you get 64 possible codons. That's enough to code for all 20 amino acids and some extra signals like start and stop. So codons are like 3-letter words, spelling out a protein. Some codons have a special job. For example, the start codon, AUG, says begin building here. It also codes for amino acid methionine. Then there are stop codons, the UAA, UAG, and UGA. They tell the ribosomes, stop building, release the protein. Quick check. What do you think happens if a protein has no stop codon? You're right. It keeps growing, becoming useless. The protein might not work if it doesn't stop at the right spot. So how do we know which codon codes for which amino acid? Scientists map it all out. Let's try this together. Look at your chart. Which amino acid does UUU code for? And what about UGA? Perfect! Notice that many amino acids have more than one codon. This is why the genetic code is called degenerate. It's redundant, which helps protect us from some mutations. Now, let's connect this to what codons actually build: amino acids. What are amino acids? Exactly. Amino acids are building blocks of protein. There are 20 standard amino acids. When your ribosome reads the mRNA or messenger RNA, it links to the right amino acids in the right order to make a polypeptide chain, which folds into a working protein. So, codons, amino acids, proteins, then U. Let's put it all together. This is where the translation occurs, and this is how it happens. First, mRNA brings the instructions from the DNA. Second, the ribosome reads the codons, 3 letters at a time. Third, each tRNA carries an amino acid and has an anticodon that matches the mRNA codon. Fourth, ribosome links the amino acid to build a polypeptide. Here's a quick question. Why is it important for the anticodon to match the codon? You're right. Even if it's wrong, the wrong amino acid goes in, and the protein might not work. Even one wrong amino acid can mess up a whole protein. Before we wrap this part up, remember these 4 big features of the genetic code. It's universal. Almost all living things uses the same code. It's non-overlapping. Codons are read one by one, not reused. It's degenerate. Some amino acids have multiple codons. It's unambiguous. Each codon only codes for one amino acid. Question: why do you think redundancy is useful? You're right. Some mutations don't change the amino acid, so it helps protect us. Let's talk about mutations. Who can tell me what's a mutation? Right, a change in DNA sequence. It can change a codon, which might do nothing, or silent mutation, change an amino acid, or missense mutation, create a stop codon too soon, or nonsense mutation. A famous example is sickle cell anemia, which is caused by just one codon change. One tiny switch equals to big effect. Okay, let's practice reading the code. Decode the sequence with your codon chart. The mRNA are AUG, GGC, UUU, UGA. So the questions are, what's the start codon? What's GGC? What's UUU? And UGA? Before we finish, let's see what you remember. Please take out any kind of paper and answer these 4 quick questions. Don't worry, it's short. The questions are, what is the start codon and which amino acid does it code for? Name the 3 stop codons. Decode this mRNA, AUG, CC, ACU, UAA. Write the amino acids in order. Explain what might happen if a codon mutates from UAU, tyrosine, to UAA. Pause the video and answer. Work quietly and use your codon chart. Alright, pens down. Let's check your answers. Today, you learned how the genetic code works, how DNA turns into mRNA, how codons spell out amino acids, and how ribosomes read the code to build every protein in your body. It's like a language that all life shares, from humans to bacteria to plants. Great job today, everyone. You cracked the code.